Uh, all right, welcome everybody. And it's my great pleasure to introduce our speaker today, Dr. Crystal Zuniga. Dr. Zuniga got her undergrad degree at the Polytechnic University in Mexico, followed by a master's in engineering and her PhD from the Autonomous Metropolitan University in Mexico City. And then she was a postdoc or a project scientist at UC San Diego, working in computational biology before she started her faculty position at San Diego State University, where she's currently an assistant professor, kicking a lot of butt and making us all look bad because she's been doing such a phenomenal job. Her lab is super duper active, and she has been a recent recipient of several big grants, including the BRC Bio grant from the NSF, as well as the DOE's version of Early Career Award, which she recently received. And her team works in metabolic modeling, and about which she's going to tell us a little bit about today. So without much ado, take it away, Crystal. Thank you.
Thank you, Arun. Yeah, today we're going to talk about how we are doing modeling of C1 metabolism. But before doing that, I want to tell you why we are doing this. So there is a big boom on the bioenergy, bio everything. And then and the reason why this is happening is because the White House had these big bold goals for the US biotechnology and biomanufacturing, in which they are highly bold. Like in 2023, they want to accomplish these goals, which have we all in certain way we're working towards them for all the people in the biotechnology area, in which it is very challenging, but in 20 years, we want to demonstrate cost-effective, sustainable routes to get rid of bioplastics. In food and agriculture, we want to reduce methane emissions. And then in the supply chain, within the next 20 years, uh, we want to produce at least 30% of the US chemical demand. And in the next 20 years, we want to increase the manufacturing scale and cell-based therapies across different diseases, and also increase the manufacturing process by tenfold. And then there is also cross-cutting advances, and this is one of the most bold. It looked to me, we have been working a lot on identifying new functions. So a lot of you are familiar with hypothetical proteins or non-functions, all these kind of things. So the goal is that within the next 5 years, we are going to sequence the genomes of 1 million microbial species and understand the function of at least 80% of the newly discovered genes. That is highly challenging, and that is what the all the framework for this talk, because actually we have been doing that for a while, but now it's time to really apply it at large scale. So in my talk, we are going to talk a little bit about the principles of metabolic modeling as a tool to understand the metabolism, and also the applications, like why we use this modeling for the understanding of algae, and then also unraveling intracellular interactions and crosstalk of organelles, and then a little bit about methanotrophic metabolism too. So I don't need to convince you, because you are in the right place, bioinformatics, but computational biology together with bioinformatics are one of the most growing careers in the last decade. And this is because the metabolism is so complex that we need to add in the computer to try to understand it. And there are like a different goals associated with this, is like understanding, but also visualizing, and then also give the maximum use to all this big data that is being produced currently. Uh, Genome scale metabolic models are two things. They are a knowledge based, which is just a database of what we have, and also they can be converted into a mathematical model that allows you to predict phenotypes. So, for example, to build a genome scale metabolic model, you can start generating a draft reconstruction from genomes, annotated genomes from isolates or from metagenomes. And then we use different pipelines to create this initial model. There are also different toolboxes available for MATLAB and Python that you can use to do this step. And after that, we move to the part of what is a knowledge base. And then we have this network that we curate manually using different information from databases, primary literature, uh, also the most known metabolism databases like CARE, Swiss Pro, Metaseq, and then at the end we have a highly curated model. And what does this mean, having a highly curated model as a knowledge base? It doesn't mean that like having a table that has a couple of different tables that have like different reactions IDs, reaction names, equations, and then we have this connection to the genes that allow this reaction to happen. We also categorize each pathway within this. We categorize the reactions that happen within each pathway, and also within different types of organelles for eukaryotic organisms. We know a little bit about the reversibility of those reactions, and then the next these lower bound and upper bound are like a modeling reactions that would allow us to generate certain flags through this whole network. And then we also have easy numbers. We have some notes and references of the different activities that has been reported. So in general, what we created in the lab is just these big knowledge bases that are very reliable. So usually, every time that we build a new model, we are able to annotate like anything from 100 to 500 new functions that normal annotation techniques don't pick up. So all this manual curation and association of these genes to the specific reactions and looking into, based on the genome, what reactions are possible to happen is what it really builds up this knowledge base. At the same time, we have for every single metabolite here, every single equation, we have metabolites that make up a new list also of metabolite IDs and metabolite names, and then we have different identifiers for these metabolites. So in general, this knowledge base can be converted into SBML, MAT, JSON, XLS, and then you will have a very nice and comprehensive database that you can look at every time. And then what is important because you can have metabolites in gas and soluble forms. You can look into all the amino acid metabolism, organic acids, lipids, carbohydrates. Some of the models also have specialty metabolites, hormones, photons, xenobiotes, And then you can also look into the reactions that need any single one of these metals or ions, and you can perform simulations about them. So if within your lab you had been tasked to look into the response to nitrogen limitation, to excess of phosphorus, iron. Iron is a very interesting one for pathogens. So we have been doing some interesting simulations about iron availability and things like that. So this knowledge base actually lets you do all these type of simulations. And then when we convert the model into a metabolic model, so we have this very comprehensive network that we created, and now we want to predict things with that. So this constraint-based mathematical modeling allows you to organize the metabolites and the reactions in matrices that are connected to these boundaries that I showed you before, and allow you, using flux balance analysis, to calculate what would be the flux through this whole network. And that it is a proxy of different types of multiomics. So just to let you know, what we want is like we would like to have a model first, and then we can have a proxy of what will happen in the different conditions with a flux, and that will give you to do experimental design more effectively. And that this is how the model architecture allows you to introduce all these kinds of data sets. So you can introduce target metabolomics to have a specific stage of the cell. You can also include transcriptomics data, proteomics data, and metabolomics data, because we have all this information within the models. And then you can also include different constraints, which is physiological data. If you have any measurement of nutrients uptake, growth rates, if you have a knockout, a knockdown, if you have secretion rates, for example, for human and eukaryotes, it is very common that they measure lactate secretion, sucrose uptake. These types of things are very common to have. So then the models allow you to contextualize all these kinds of data sets. And then how does this happen? So by having a simple glucose uptake rate, methane uptake rate, you can simulate how is the flux through all the metabolism, a lot of metabolic reactions, but maximizing for this biomass objective function. And this biomass objective function contains what is the percentage of the metabolites that make biomass. So you can have lipids, amino acids, nucleotides, etc. So then you can include all these data, omics data, as I mentioned, and then by changing this biomass objective function, we have been able to fully characterize what is happening within this network. But this is not new to you. So you live within networks all the time, and you have been using these networks for a long time. And this is an example. An example of a network, it would be like the map of San Diego, right? And then if we want to produce any biochemical, for example, if you will want to go from something here to all the way to San Diego State University. So you have a goal. And once you have a goal and you have a network, you can add into it live data, which will be the multiomics data, right? And it will inform your decisions to what route to take. So this is kind of the underlying thought behind metabolic networks. So once you have a goal and you have a network, if you can inform this using any live data, you are going to be able to travel faster and then maybe sometimes more efficiently. And what happens is that when you translate this to metabolism, a new field came up, like a metabolic engineering, in which you can inform the decisions of what paths to reduce or enhance to increase this product of interest. But how does metabolic network really look like? You can see here we have this network, which is going to be the knowledge base, what I show you. And then you have in circles metabolites, and then each arrow represents one reaction. So as you can see, at some point, for some processes, it is easy to follow one by one, but there is a point in which there are so many connections among things that is very hard to do it. So this what you are looking at here is an Escher map, and this Escher map it is a web-based tool that allows you to jump in and draw all these metabolic reactions based on the model by doing it very easy, very quickly. You can just go click, click, and it keeps suggesting you networks based on this. So you can also add your multiomics information here, either any gene-associated multiomics information such as RNA-seq, proteomics, or metabolite levels. And then what this is what we call flux distribution, in which you will have certain numbers. Maybe it's too small, but you can see some numbers associated with the names of the reactions here. So that is what we call flux distribution. Just to give you an idea, we have been doing this for different organisms and with different goals. A lot of them focus on bioenergy, but also for some pathogens. And lately in the lab, we have been doing this for wastewater treatment, for the characterization of different photosynthetic organisms. We are highly focused on the production of pigments using different algae, and also looking at cell-cell interactions of algae and fungi, and cyanobacteria and yeast, bacteria, and aspergillus. So this is the next step into these metabolic models, like how we can understand the two or more organisms going together. And then for this, just to show you an example of how we have been using these models to improve annotation and to go towards the this last goal of improving, learn more functions about the genome. We did this. We have, using the model of Chlorella vulgaris that you see here, we wanted to understand and to characterize the transporters within the cell. And this is important because we have certain number of annotation for some organisms. For microalgae is highly poor. Something between 20 to 80% of the functions are hypothetical proteins. And this is not surprising, and it's also true for E. coli, which is the most studied organism. Just the last number that BioSeq released not long ago, still 15% of the genes we don't know anything about. 15% out of 3,000 roughly genes in the E. coli genome we don't know anything about, and 20% they have a partial annotation. So that is the most studied organism on Earth. So for other organisms, it's more challenging to understand these functions. But we are also failing on translating what we know from the original ones to others, right? And these models are like the connection between these. We can be used for that connection, but also they can be used to do to predict new functions based on the fluxes. So you have this kind of model. Let's think about this cell as Chlorella vulgaris, a green algae that has a mitochondria, it has chloroplasts, it has peroxisomes, it has different organelles within the cell. And what happens is that during the time that we build the model and we do manual curation, there are some reactions that we need to add into the model without gene just to enable the function of the model. So then we have these reactions that they don't have a gene, but in order to make sure that there is connectivity and the flux distribution can be predicted, we add these reactions. And these are called orphan reactions. So these kind of orphan reactions are very common in different eukaryotic models. And this happens in the model of Chlorella 2. So when we have the model, we can predict flux distributions and different conditions. Heterotrophy, which is dark with an organic carbon source, and photoautotrophy, which is using CO2 and light as unique carbon sources. So you can see here that in one condition, some of those reactions are active versus the others. So this allows us to have a better picture of what is going on within this organism. So what we did is we collected targeted metabolomics of the biomass, meaning breakdown of protein, like we measure all the 20 amino acids, we measure different kinds of fatty acids, we measure nucleotides and carbohydrates over time for those two conditions. And then what we did is we had predicted flux distributions for each one of these time points. And then, as you can see in this graph, you can see that this time point at this more than 50% carbohydrates will give us a point here for every single reaction in the model. So the model has approximately 2,000 reactions. So we have this information about flux every time for each biomass composition. So then we were able to generate like trends of flux distributions under heterotrophic growth and under photoautotrophic growth. But at the same time, our collaborators collected gene expression data over the course of time, in which, under the same conditions, nitrogen is depleted and the algae are growing and keep growing even though nitrogen is depleted, but then they start accumulating lipids and carbohydrates. So by looking at these dynamic behaviors using flux distributions and RNA-seq sequences, we were able to retrieve trends over time in two different data sets. So, as you know, in the model, we only had 981 genes. But when you have the whole on any RNA-seq data set, it gives you a very good picture of all the activities happening there. So in this case, we got reads for 7,100 genes, which means that we are only recapitulating around 15% of what is happening in the model of all the total possibilities, right? So, because we had these orphan reactions that I showed you before, that they didn't have gene, but they have flags, now we wanted to see, can I connect these reactions that I have a flags trend with compare them across all the trends that are in the RNA-seq data set, and then find a possible function for them, right? So when we did that, we created a comparison, and then we were able to annotate over 100 new genes. So what happened is that every time that we had a trend for an orphan reaction that matches that trend in this data set of RNA-seq, we retrieve the gene and then we reannotate the gene. And this just happens 2 years after we finished the model, published the model, and then we did this study, and then we used this tool that is called Interpro that gives you annotations for those genes, and we were able to find these new annotations for more than 100 genes that we confirmed. Because out of the analysis, we were able to obtain around 250 hits, something like that. And then when we rerun the annotation and then confirm that this activity was something similar to what the model was predicted, right? The flux distributions. So then we went and annotate this into the model. So reactions that in the past didn't have a gene here, now we have a new gene. And interestingly, these happen across all the different organelles. So we annotated 2 in the glycosome, 3 in the cytoplasm, 17 in the mitochondria, 18 in the chloroplast. And yeah, so we had all these new possible functions within the cell. And just like that, we increase like in 10% the knowledge of the new annotations within the model or the metabolism of Chlorella vulgaris. And what happened is that also there is no one protein, one gene. There are some multi-subunit enzymes. They are also different genes that can code that same. We also look into those, but as you can see here, like purine metabolism, we were able to annotate several reactions highly conserved, across the cytoplasm, the chloroplast, and the mitochondria, and the glycosome. And then in the glycan biosynthesis, we were able to do a lot of annotation in the cytoplasm, for some of the fatty acid and transporters into the chloroplast. What happens is that cells start producing drops of fatty acid or of lipids within the chloroplast. So all this metabolism happens only there. Some of the reactions also, all the metabolic reactions only happen here. So as you can see, there were these orphan reactions across the whole the network, and we were able to increase this knowledge successfully. And in this figure on the left, you can see all these highly interconnected, like lipids synthesizing here in the chloroplast, and also lipids in the cytoplasm. Then part of the glucolysis ending in the cytoplasm, and then bringing in things into the mitochondria. And there are like all these processes happening at the same time. So now we are able to target new genes that can be annotated within it. And then just for this first part is like the models are very good at contextualizing data. Targeted metabolomics data is their favorite. We love having this type of data. Now here in the lab, we can produce pretty much all these kinds of data using different analytical methods. And then once we have these broad predictions, we can also combine them with multi-omics data, and we can characterize the crosstalk of organelles, and then characterize new gene functions. And then within the scope of this work, we increase the, the number of known functions of chlorella, and that was also very good. And then just to show you, now in the lab, we have Chlorella vulgarius and different types of algae growing here. And then we are now using all these properties of algae to create foods. And then this is one of the projects that we have here in the lab, in which we are very good at knowing when is this high protein content, high lipid content of the algae, that using the models we can actually supply the only amount of nutrients necessary to keep that nutritional profile. And then what happens is that starts here, in which we do the modeling, we form the experiment, and then our collaborators have been very active and characterizing all these nutritional properties. They have developed a product that you may have heard about, this algae space WAC. And right now we are evaluating the quality, and then they are doing the sensory analysis. We have some samples, and eventually we would like to send it to space. And this is important because not only in space, but on Earth, increasing the nutritional properties of food will have a remarkable effects on the human population. And currently in the lab, I'm going to tell you, this is pure work that has been carried out here at CSU, in which a talented team has been working on the reconstruction of the model of Dunaliela salina. One of actually your teammates, Jenna, she took the lead on this, and for the last 2 years, she has been building this model. And now it is ready to go. So in which we characterize the protein, lipids, pigment, glycerol, carbohydrate, and we are exploring new culturing methods. So we can see here that the algae changes remarkably its profiling, and then you can also see how the protein content is high in the different algae. Also, you can see that there are certain amino acids that are richer in the different Dunaliella strains. And then, yeah, so we have now fully characterized the fatty acids and other metabolites. So just to remind you, we had these biomass objective functions that we always love or want to characterize. We have done this over the course of growth of Dunaliella, in which Dunaliella passes from green to yellow to orange. And then we are capable to predict these flux distributions and start making assumptions of what would be the best conditions to grow this microalgae. We have also performed different phenotypic analyses that have enabled us to um, test different kinds of culture conditions that potentially can increase the yields of algae to make it better. Uh, um, on our collaborator side, they had been characterized the odors of this algae because the odor is 70 to 90% of what you taste, something like that. So they have been characterizing what are the odors of different algae that we have. And then this is an example of the space walk. There are some strains that are very colorful. So we are working on reducing the green, but I mean, in general, the guac is very tasty and it has a very good flavor. Um, the other thing is that it's very surprising because they were able to manage dehydrating and reincorporating. So when they dehydrate the avocado and have all the components dry and then you just add water, um, to me it tastes almost the same. Yeah, this is just very interesting. And this is one of the applications of the that we are having now in the lab. Another thing that is also is like that this allows and the modeling allows for accurate technology development. And this was part of still of my work back at UCSD, in which they we work with this when wastewater, in which they have this very big response of algae, in which having algae culture at these larger scales, it becomes not sustainable sometimes because of the cost of the nutrients, right? So that's why a lot of options have came up, like using wastewater or others. But to have more control, what we did is that we used these simulations. We collected data, then we used these simulations, and then we calculated how much was the only strict necessary nutrient, like in the form of nitrogen, to keep the cell in the stage that we wanted. And then in this way, you could actually control this composition and maintain the cell in the state that you wanted with like less than 10% of the cost of the nitrogen supply. So this is another application that when we combine models with technology, you can have very good results. Yes, so just here in the lab, we have this model of Dunaliella salina. We can produce pigments now. We're also working with other algae like Hematococcus that produces red pigment. We're also working with diatoms, which are marine algae, same as Dunaliella, which they don't compete with freshwater crops. And then, yeah, so we can enrich the nutritional properties of food, and then we can control the physiological state. Another project that we are looking into about C1 metabolism, it was the development of the metabolic model of methylacetyphilum fumariolicum. And this metanotrop was very interesting because it is very interesting because it can grow at high temperatures above 50 degrees. And then for this, it is usually very interesting because the um, the industry wants microbes that can tolerate higher, harsh or conditions which are comparable to chemical. So then that is very interesting. We perform some simulations, identify where the bottlenecks, but we have also done Rhodopseudomonas, Clostridium, Nitrosomonas, and other models. And for this, just to follow up on what Arun was saying on bioenergy and this grant that DOE kindly awarded us we're gonna build the next tier of metabolic models in which we are going to pass from what I showed you today was metabolic models by itself. But now there are new trends in metabolic models that you can convert them into transcription and expression models. And these models are more complex and they allow you to include the genome sequence. So we can start performing simulations about changes in the genome sequence. How would that affect the phenotype of the organisms? These new models can be expanded to pH models and temperature models, which is gonna be very critical in the next years because despite the fact that we had been awarded for several years about the effects of the climate change. Right now, we are really looking into it into more extreme temperatures. So we will like to have more resilient microorganisms that allows us to be prepared for these extreme conditions. So we believe that these models are going to play a critical role in trying to understand uh, these mechanisms and how to be ready for them. So this is what we're going to be doing in the lab within the next 5 years. And with that, I just want to say thank you to the collaborators. So Mike was always the lead of the projects, and Maciek, he produced all the biomass data that I show you for the understanding of genome annotation. And then we collaborate with Dr. Sao and Liu here at the CSU. They are the ones developing the food and the space work. And then, yeah, and then to the whole team in the Zuniga lab, we have a very talented postdoc, and we have Jenna, who is building the model of Donaliela. We have Ayla building model, and different people involved in the culturing of the algae. Yes. So just like a final concluding remark is like that the 21st century has been called the century of the biology. And then the biology and the computational biology, I will add, and I think that we are going to be or have a lot of support to attain great discoveries. And at the same time, we will be developing transformative technologies that will make this bioeconomy possible at all the different levels, biomanufacturing, bioenergy, and different things. So with that, I will open the floor if you have any questions. And I just have one announcement, like we have some projects open. Yeah.
All right. Yes, that's always good to know. We have students who are definitely looking for projects. Yeah, so we do have some questions. Actually, I thought I didn't really understand until you actually said it, the joke about space guac sounding like spacewalk. So I don't know if that's what you meant, but I was like, oh, space guac, spacewalk. Got it. But yeah, we have some questions from the students. So, oh, and by the way, everyone had to run to a seminar. So that's why he's not here right now. So one of the questions was, how accurate is the mapping of these pathways, especially considering like the experimental changes? And I think that means you had to, use experiments to get the mappings, but if those experiments were a little wrong, could that mean that the mapping is also wrong? How accurate are they? How do you know?
Yeah, I mean, we run like a t-test, homogeneity test on the trends, and then for that we compare. So we fit different models like linear, exponential, and then for every one of them we compare the coefficient that was calculated, and then we decide if it's the same or not. We have a little bit of that, but also yes, if this data, you mean if this data the one that I used to make that
Well,
the flag distribution?
it does seem like the way you were writing about it, you're talking about it earlier, you had a metabolic maps that was like street maps, right? And are you assuming that those maps are accurate? Does it rest on that assumption that they're extremely accurate?
Oh
Or
yes,
it does. Okay, so if they were, and how certain are you of the accuracy of those input models?
highly certain because we review reaction by reaction.
But you didn't make up those maps.
Yes.
You did. You made those maps yourself. Oh, okay. Okay. So you already know how the maps are before you did it. Got
Yeah.
it. Got it. Okay, cool. That's
Yeah.
good to know.
This implies a lot of manual curation,
Okay.
a lot of checking and reviewing. Yeah.
Cool. Um, how did you find, another question was, how did you find that the predicted flux trends and gene expression trends was found for 65% of multi-subunit enzymes and 79% of allosteric reactions?
Yeah. This is how well predicted they were because we could make the comparison between the the prediction with the RNA-seq based on the reactions that we already have gene for, right? So there are a lot of reactions in the model. We have all these reactions in a model. So let's say only these circle we don't have genes, but for everything else we have. So then when we compare these ones that we knew already based on what the trend here, right? So for those we know we have a gene and we have a reaction. So for those we were able to calculate this, in which there are some the flux that is predicted versus the RNA-seq for this that we knew the gene are able to resemble 65% of the multi-subunit enzymes and 75% of the allosteric reactions. So there are these genes that have multiple. There are these reactions that have multiple genes. So then maybe I have one of those here. So here, this is an example. This one, for example, this one can be an example of a multi-subunit enzyme. What you see in 137. So this gene plus this gene will make these flags, right? So we have a single flag, and then we have all these expression for these genes. So then for 75% of them, we were able to predict like the flags change at the same time that the gene.
Hmm,
Mhm.
that's great.
Mhm. Yeah, it's very powerful tool in
Yeah, that's
general.
really powerful. Um, so I had a question about whether your model is accessible to everybody, and
Oh,
why did you have different model formats?
I mean, because if you like Pythons, people like Cobra Pi, so you use JSON. If
Got it.
you like MATLAB, you can have math, and things like that. But what I want to show you is, yes, we actually have all the models. If you go to my website, which is CC Lab, I'm gonna drop it in the chat. You can see all the
You have them all available.
yeah, and
That's
also
great.
in the um in the papers we always publish the model. Or in the Git on my GitHub.
That's good. Question is, how might the construction of previously unstudied metabolic pathways affect our understanding of the structure-function relationship in proteins?
Or previously unknown.
Or yeah, I don't, I'm reading the question. I'm not quite sure, but the question is, how might the construction of previously unstudied metabolic pathways?
Oh, that is a good one. Yeah. So we have that challenge all the time, and more tools have came up. There is this lab of Costas Maranas in Penn State and Bacillus Hatziniumycatis and Switzerland. What they had come up with is like a machine learning model in which they input all the possible activities of the by eating numbers. So if you give them what molecule you want to start with, and then what molecule you want to end, or what molecule you want to produce, they will be able to give you a suggested pathway.
Oh, that's cool.
Yeah, that is very interesting and very helpful, because when we're building the model of citrus, which is about to come out, plants have such a complex metabolism and they are capable to produce a lot of different things. So yeah.
Yeah, that's right. I mean, one thing I noticed about plants, when I studied beetle plant interactions or insect plant interactions in ecology was the amount of what they called secondary chemical compounds that did not seem to be useful to well, I mean, they could have been useful to the plant or they could have been byproducts, but a lot of the ecologists tended to consider them plant defense compounds. And it seemed like they could have lots and lots of these things to protect themselves because, if you're a plant, you can't run away from your predator. So you have to sit there. And so like the only way you can really defend yourself is chemically or physically. You
Yeah.
can have spines, or you can have toxins, or you can have terpenes, you can have bark, or you could have like nasty, nasty chemicals. And so it seemed like plants were able to produce all these things, at different stages of their life under different conditions, more than, any animal would do. And
Yeah.
somehow the cells did just fine. I don't know if they had to have extra protective mechanisms or something, but is that what you're talking about? That there's all these like crazy chemicals out there?
Kinds of
Yeah.
metabolites. And then to be able to, of course, what you see here is that the first manually curated model, it was reaction by reaction.
Mm.
So it's in the same way that we will build models for new pathways.
One at a
So
time. That's really tedious.
it
But once
is,
you have
but
it, you have it.
exactly. So
Yeah.
once we have it, like, for example, the E. coli model, it has been cited like 13,000 times, the first
Oh
one.
yeah.
But
Once you get it, yeah.
since then, it has been published every single update on Nature Biotechnology and having
Cool.
the same impact. Like, it's just so once you have the first one is the having the first one is the challenging one.
Right. Yeah. Yeah, they don't pay people enough. I mean, they don't give enough credit for that type of work, in my opinion. There's people who just spend, their careers mapping down pathways. And then the people who use the pathways often get, like all the credit, right? But if you don't have that, what are you going to do? Sometimes as scientists, we're so excited about the answer, but we have to appreciate that someone took the time to map out every single gene and every single metabolite or every single, or count all the different species there are, or whatever it is that they did. That stuff is
Mm,
so valuable to every researcher moving forward, and it's just to me it's underappreciated sometimes. But
yeah.
it's something I can't personally do. I don't have the patience for it.
But the people that they do, I mean, like, once if you something that you like having perfect lists of things and finding things, because there are people that like doing that. I
Yes, they
mean,
do like that, and they often have trouble getting funded because it's like it's not always hypothesis testing. Sometimes it's just like factual fact and information generation,
yeah,
but it's so important, and some people really like it, and I'm, good for them. It's really important stuff.
yeah, it's what we do a lot here in the lab,
Sure.
and also the fact that we need to put it together with something exciting.
Absolutely. Yeah, yeah. Like I do some metabolite stuff, and we need someone to figure out what all the metabolites are. Or it's a basically unknown. We don't know anything about it, and we can't tell you if we find an interactions. I can't tell you anything because I'm like it's unknown, until someone discovers it. Yeah. Let's do one more question then. For metabolic engineering, can you explain the process of engineering a system to overcome a thermodynamic bottleneck? Like, which genes do you knock up or down to do that?
Yeah. We don't have any example here. The thing with chlorella, and it's why we are getting away from it, kind of leaving it behind, is because there are no genetic tools for it.
Oh, yes, right. Right. Yeah.
So it is very hard, but
So you can't knock things up or down or knock things out. Yeah.
very
It's
difficult.
chlorella is very difficult. Yeah.
Yeah, but there are other algae like a phylactylum that has all the CRISPR ready to go and things like that. So, for example, in this one that you can see, the models allow you to see the flux, right, in one condition versus another. And what you can do is, if, for example, your product is ethanol, there are different algorithms in which they tell you, okay, you should knock down this one and it will go through here.
Hmm.
And then on the process, you maybe synthesize something else. So there are all these algorithms that allow you to calculate, to suggest some knockdowns within the pathway, depending on the condition that you are growing your microorganisms and so on. So these algorithms will do is take your network and then say, okay, maximize for ethanol synthesis. So then based on that, they will play with different flux distributions and then say, okay, you can cut this out, you can cut this out, you can cut this out. It will suggest different knockouts.
Hmm.
And there are different algorithms that do that very accurately. So this has been fully tested in different bacteria like E. coli, Bacillus. There are a lot of different good examples of metabolic engineering, even in cyanobacteria. Yeah.
Hmm. Can you
So
insert, I assume you could insert engineered genes. Do you think this one's going to be more stable or faster or
yeah.
better at binding, and then just knock that and just, insert that genetically and just splice it in? Yeah.
Yes,
Yes.
definitely.
Yeah.
In fact, we're actually doing that for a new, but it's not new. It's called Simomonas mobiles for the Great Lakes. I became a member of one of the centers of UE, and what we're doing for them is building the model of Simomonas because they have all these heterologous pathways that they have engineered that they don't only want to produce ethanol, but they also want to produce isobutanol,
Oh, wow.
which is creating like a new pathway here.
Hmm. Hmm.
So so we want to create that model and to be a very robust model because what they want to test is different carbon sources. because they come from the lignocellulose, like from the bioenergy crops. So they digest the poplar or whatever tree, and then they have this mix of different substrates. So we want to look into that using the model of an updated model. So we are going to be working for them doing that here in the lab.
Oh, there's another question I saw in the Q&A. Sorry. What are the most important pathways for constraint-based modeling?
I mean, as usual, if you have an heterotrophic organism, the key pathways are going to be the glycolysis, the TCA cycle, right? And then if you have another organism with different metabolism or metanotrophs or others, probably the most important and the reactions that should be the most connected ones, are going to be these ones, that the starting points that you know. But then you have modeling of human in which you use such a complex media, right? So then it doesn't become a simple just give them methane or CO2. It becomes a set of 20 to 30 carbon sources that you need to, do it at the same time. So the more information you have, the better, because but yeah, it will depend a lot on the capabilities of your organism.
Of the organism. Right. Yeah, the complexity, apparently.
Yeah.
Yeah. Very cool. Well, thank you so much for your time and showing us your amazing work. And like your lab is just enormous. Like, do you even have space for all those people?
A
It
lot
must
of
be
them
get
are
tough, right? What?
remotely. And we have a space, we have a computational space, and then, yeah.
Yeah, no, it's good. That's great. Well, thank you so much, Crystal.
Thank you.
Hope I get to talk to you again soon in person. But wonderful stuff. Wonderful stuff. All right. Thanks everybody for attending. I will, this is
Thank
recording
you.
if you, or this has been recorded if you ever want uh, to have it for any reason. So, okay. Bye.
Bye.